Today, we're going on a treasure hunt. But forget gold or ancient artifacts. We are searching for something way more valuable, and it's hidden in plain sight. I'm talking about the powerful chemical compounds locked inside the plants all around us. So, let's get started.
Okay, so this slide right here, it basically sums up our entire journey. On the left, you've got a simple piece of cinchona bark. But after this whole scientific quest, you can pull out the specific, powerful molecule you see on the right, quinine, a compound that literally changed the history of medicine. So the big question is, how do we get from a piece of bark to a pure, potent chemical like that?
Well, these treasures we're after have a scientific name, of course. They're called phytoconstituents. It's really just a broad term for the incredible, vast array of chemical compounds that plants produce. They're the reason a plant has its color, its scent, and, a lot of the time, its medicinal power.
And you know what? We run into these treasures all the time. The caffeine that kickstarts your morning, that's one. The curcumin and turmeric that gives curry its vibrant yellow color, that's another. Or that cooling menthol in mint. These are all phytoconstituents that scientists have found, purified, and put to good use.
Now, every good treasure hunt needs a map and a solid set of tools. For a phytochemist, that's a plant chemist, the map is a clear three-step process that lets them pinpoint and unearth these amazing molecular treasures. So the whole process really boils down to three key stages. First up is extraction. That's all about just getting the raw chemical cocktail out of the plant. Second, you've got isolation. This is where we zero in on our one target, our treasure, and separate it from all the other stuff. And finally, there's analysis. This is the crucial last step where we confirm, yep, we found exactly what we were looking for.
The best way to think about extraction is to imagine you're brewing a pot of coffee. The coffee grounds, that's your plant material. The hot water, that's your solvent. The water runs through the grounds and pulls out all the good stuff, the caffeine, the flavor compounds, and leaves the solid grounds behind. Scientists do the exact same thing, they just might use different solvents, like alcohol, to pull out different kinds of treasures.
Okay, now for isolation. Scientists have this fantastic tool called chromatography. Imagine a foot race where every single runner has a slightly different top speed. Chromatography basically sets up a racetrack, and as the chemical mixture starts moving, all the different molecules separate because they run at different speeds. The fastest ones get to the finish line first, which lets scientists collect each one all nice and separated. It's pretty genius.
All right, enough theory. Let's put this toolkit to the test, shall we? We're going to go on a very specific hunt for one of the most recognizable plant compounds in the world, that cool, refreshing sensation we know as menthol.
So our starting point is the peppermint plant. Its official name is Mentha piperita. We're going after the one specific molecule that's responsible for that signature minty coolness. That is our target treasure.
Let's dive into step 1, extraction. What you're seeing in this diagram is a method called steam distillation. The boiler on the left forces hot steam through all the peppermint leaves. This steam is hot enough that it actually bursts the plant cells and releases all those menthol-rich essential oils, turning them into a vapor. So now you've got this hot steam, and it's carrying the essential oil with it. It travels up and into that coiled tube, which is called a condenser. Cool water is circulating around the outside of that tube, and it makes the vapor inside cool right down and turn back into a liquid. And because oil is lighter than water, the peppermint oil just floats right on top. Super convenient and ready to be collected.
Now, here's the really cool part, no pun intended. The isolation step from menthol can be surprisingly simple. As that peppermint oil we just collected cools down, the menthol molecules start to find each other and stick together, forming these beautiful little white crystals. They literally crystallize right out of the oil, separating themselves from everything else. It's just a really elegant bit of chemistry.
Okay, so we've got these white crystals. They look like menthol, they definitely smell like menthol, but in science, that's just not good enough, right? How can we be absolutely 100% sure that we've found our pure treasure? Well, this brings us to our final step.
And step 3 is analysis. We are back to our chemical race, just on a much smaller scale. This is a technique called thin layer chromatography, or TLC for short. We take a tiny spot of our dissolved crystal and place it on the starting line of this special plate. Then, the whole plate goes into a jar with a bit of solvent, which acts like the starting pistol, kicking off the race up the plate.
And here we have it, the photo finish. As the solvent creeps up the plate, it carries our sample with it. Now, because of its specific chemical properties, pure menthol will always travel a very particular distance, creating a single clean spot. To be sure, scientists run a sample of known, pure menthol right next to it as a control. If our spot travels the exact same distance and lines up perfectly with the known sample, boom, we have confirmation. Treasure found.
This three-step process, extraction, isolation, and analysis, it's not just for getting the coolness out of mint. I mean, this is a fundamental technique that has unearthed chemical compounds that have truly shaped human history.
What's absolutely incredible is how this process can turn something dangerous into something beneficial. Take atropine. It's an essential medicine used to regulate heart rate during surgery. And where do we get it? It's hunted for and isolated from the famously poisonous deadly nightshade plant. A poison becomes a lifesaver. It's just wild.
And hey, remember this one from the beginning? Quinine, isolated from the bark of the cinchona tree. This was the first effective treatment for malaria. It's a discovery that saved countless lives and allowed for exploration into parts of the world that were basically uninhabitable before. And sometimes, the hunt leads us to treasures hiding right in our own kitchen cabinets. This is curcumin, the active compound in turmeric. Using this exact same process, scientists can isolate it to study its potential anti-inflammatory and antioxidant properties for modern medicine.
So, at the end of the day, this is so much more than just an academic exercise. This scientific treasure hunt is a vital engine of discovery. It takes clues, sometimes from ancient traditions, and transforms them into standardized, pure compounds that become the medicines, the flavors, and the materials that define our modern world. Which leaves us with one final, just fascinating thought. There are millions of plant species on our planet, and scientists have only just begun to scratch the surface of the chemical treasures they hold. It really makes you wonder, what future cure, what amazing new flavor, what world-changing breakthrough is still out there, hidden away in a leaf or a root, just waiting for the next treasure hunter to come along and find it?